Analytical quality by design, it is also called as AQBD, and it is for the method development for the analysis of pharmaceuticals. As we know, quality by design approach is a risk-based approach for the development of the pharmaceuticals formulations and also for the development of analytical methods. It involves understanding and mitigating the risk and having control strategy. Also, QBD is for the life cycle approach. In this video, we are going to learn regarding the analytical quality by design. Quality by design principles are not only applicable to the formulations or drug products, but these are also applicable equally to the analytical method development. Analytical quality by design, AQBD, is the application of QBD principle or principles to the analytical method development, and it focuses on the building quality into the method rather than testing it at the end. As we know that the analytical methods are required to be robust, accurate, specific, suitable for the testing of pharmaceuticals. These are required to be reliable so that the quality can be assessed and quality can be maintained for the pharmaceuticals. Quality of the pharmaceuticals is directly related to the safety and efficacy. Many of the regulatory agencies provide the requirement for the analytical methods. Regulatory agencies encourage quality by design approach adoption for the analytical methods. ICH guidelines are there like ICH Q8, Q9, Q10, Q14 for the QBD. For the analytical methods, the validation is performed as per the ICH guideline Q2 and it is developed as per the ICH guideline Q14. Mainly, AQBD is a concept covered in the ICH guideline Q14. It ensures method robustness, reliability, and regulatory flexibility. Key elements of the analytical QBD. So first is the analytical target profile. It is abbreviated as ATP. It defines method purpose and performance requirement. As like in the QTPP in the formulation development, it is ATP in the analytical QBD. Then critical quality attributes, CQS. These are the method attributes that impact the performance of the method. Critical method parameters, that is CMPs. These are the experimental conditions affecting the CQS. Risk assessment. It is required to identify the high impact parameters for method robustness. Design of experiments, that is DOE. It is a tool for the quality by design. Structured study for method optimization. It is the main aim of DOE. Analytical QBD workflow defines ATP for the accuracy, precision, range, and specificity. Performs risk assessment, that is FMEA, Ishikawa diagram. These are the tools for risk assessment. Then perform the DOE study and evaluate CMP effect on CQS. Then the next stage is establishing method operable design region. As like in design space, here it is MODR, method operable design region. So if you use some parameters from this region, you can get the accurate results. Validate method against ATP requirements and implement the control strategy and continuous monitoring. This is the analytical QBD workflow. Once again, we will review it. First is ATP, second is risk assessment, third is DOE, fourth is MODR, fifth is validation of the method, sixth one is the implementation of control strategy and continuous monitoring. Now, what are the advantages? Why AQBD is required or it is suggested? So, advantages are like it can give you the robust and reproducible methods with reduced variability, because you will have the risk, you will have the complete understanding, you will have risk assessment and mitigation plan in your hand. It will have or provide the regulatory flexibility, that is changes within the MODR may not need reapproval. Same like is design space, here it is MODR concept. Then enhanced method lifecycle management, it is as per ICH Q12. Then reduced method failures, there will be less number of OS and OOT results because of the method. Then efficient method transfer across the labs and sites. Then analytical QBD practice. Example, HPLC method development. First is defining the ATP. For example, assay of the drug substance should be within the range of 98 to 102%. Then identify the CMPs like pH, flow rate, column type, and the detection wavelength. Then run the DOE to establish the MODR, and then validate method robustness and ruggedness. So AQBD is widely used in pharma, also for the impurity profiling, dissolution, and for the bioanalytical methods. AQBD helps and supports regulatory submissions, and it is required and ideal suggestion for the global compliance for the method. This is regarding the AQBD. I suggest you to go through ICH guidelines to have the complete understanding about these topics. I will be making many more videos on ICH Q2 and Q14 related to this topic. Thank you for watching the video.